Okay, so good evening, everyone. Today we are in the week 4 tutorial session of the NPTEL course Genetic Engineering Theory and Applications. So I hope all of you have gone through the week 4 lectures. I'll start today's session. Let me start sharing the screen, today's slides. Just give me a moment. I hope the slides are visible. Yeah, okay. So sorry for the delay. Actually, I was facing some internet issue here in IT Bombay today. So So let's start with today's session. So I hope the screen is visible. Okay. Good evening, everyone. I'm Akash Jyoti Pathak, and I'm one of the PMRF TAs for this NPTEL course, Genetic Engineering Theory and Application. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So we will first quickly recap what all was covered in week four. What were the different topics which were covered, and what all did we learn in week four? So in week four, it is continuation of week three actually. So the basics of cloning part two. So here in the first there are three lectures in week four. In the first lecture we learned about the enzymes that are used in molecular cloning. So what are the different enzymes used in molecular cloning? Can anyone quickly recall and tell what are the different enzymes that are used in molecular cloning? So some of the main enzymes that are used in molecular cloning are, first of all, is restriction enzyme, isn't it? So restriction enzymes, uh, we all have learned about restriction enzymes in class 12th, right? They are one of the key players in molecular biology, and it is very widely used. Then other than this restriction enzymes, there are DNA ligases. Okay. So, so restriction enzymes come under the category of nucleases, basically. So these are endonucleases. So we will learn about these in detail. So what are the, so these are come under the class of nucleases. Then there are DNA ligases. Then there are enzymes like phosphatases, alkaline phosphatases, isn't it? Then there are enzymes like kinases. Okay, kinases. So other than this, any other enzyme which you can recall? So these are some of the very commonly used enzymes, and we need to understand what are the functions of each of these enzymes, so that when performing a cloning experiment, then the basics or the concepts of what is the function of each enzyme must be known. Uh, so that we can exactly understand how we can perform different experiments. Okay. Then in restriction enzymes, we learned about what are the three types of. So there are the three types of restriction enzymes. So what are the three types of restriction enzymes? How do they differ among themselves? And what is the mode of action of each of these enzymes? And then we learned about the vectors. Okay, so different type of vectors are there. So prokaryotic vectors, which are mainly the plasmid vectors, or they are also called the bacterial vectors. Or so if you want to clone a gene in a bacterial vector, then it is usually plasmid vector. Then for eukaryotic, there are several types of eukaryotic vectors as well that we learned, like the yeast plasmid vector, yeast artificial chromosome. Then there is baculovirus based vector. Then there is mammalian cell based vectors. Uh, Okay, so we will learn each of this in today's. We will quickly have a recap of what are these different vectors, and also other than this, what was the other concept discussed, that how we can isolate plasmid from the bacterial cell. So if we want to isolate plasmid isolation using the, so one of the most commonly used method of plasmid isolation that is followed in most laboratories is the alkaline lysis method. So alkaline lysis method is used for isolation of bacterial plasmid, right? Alkaline lysis method. So we will learn about this also today. We will quickly cover these concepts so that you can again have a quick recap and understand the concepts and solve your assignment. Okay. So let's first start with what are the different enzymes used in molecular biology. Okay. So meanwhile, in between, if you have any doubt, then you can ask in the chat, or you can directly unmute yourself and ask the question. Okay. Uh, so so there are these four different enzymes. What are the so they are the enzymes which are used in molecular biology can be grouped into four broad classes depending on the type of reaction that they catalyze. So the first one is nucleases. Okay, nucleases. We had already learned about nucleases in so exonuclease. We when in the last session when we were discussing about the different function of DNA polymerase 1, 2, 3, we also talked about clinoenzyme in brief. So it has this 3' to 5' um, hexonuclease activity. Right, so all these so nucleases can be two types basically. It can be either endonuclease or it can be exonuclease. Okay. So in the next slide, we will understand what are exonucleases and what are endonucleases. Then there are ligases. So ligases are molecular glues. They are also called molecular glues. So if you so they are used to seal the nicks, or if there is a free 5' phosphate group and a free 3' OH group, then the for this two the 5' phosphate and the 3' OH group of the two strands can be ligated by the help of this enzyme called ligases. We will learn about this enzyme also in detail. Then polymerases. So polymerases are used for what? They are used in replication. So if you want to make a copy of DNA molecules, then polymerases are used. So there are different type of polymerases in bacteria, polymerase 1, 2, 3. Then there are some modifying enzymes like alkaline phosphatase, polynucleotide kinase, terminal deoxynucleotide transferase. We learned about this enzyme in the last session. So if you this terminal deoxynucleotide transferase, in which method it was used? Anyone? Do you remember? So this was basically used to remove the nucleotides from the 3' end of a oligonucleotide. So if one wants to remove the nucleotides from the 3' end of an oligonucleotide, then this enzyme can be used. Okay, so let's learn about this enzyme. Quickly revise this the function of these enzymes one by one. So nucleases are what? As nucleases means they are nucleic acid digesting enzyme basically, isn't it? So these are nucleic acid digesting enzymes. So so you must have we all know that there are these are hydrolases, comes under the hydrolase class of enzymes. So nucleases, proteases, then there are uh, chitinases. So these are the different enzymes. So nucleases, proteases will digest proteins. Nucleases will digest the nucleic acid. There can be different types of nucleases like DNases, RNases, isn't it? So DNase will what will it do? It will digest the DNA molecule. RNAs will digest the RNA molecule. Okay. The what are nucleases? What will it do? So nucleases degrade DNA molecules by breaking the phosphodiester bond that link one nucleotide to the next in a DNA strand. So if you see this figure, so this is the DNA. This is the double-stranded DNA molecule, and what is shown over here? So these are the sugar phosphate backbone. So the sugar phosphate backbone, and what bonds are present between these? These are the phosphodiester bonds. So the phosphodiester bonds are present between two sugared units. So DNA will have deoxyribose sugar, isn't it? So two deoxyribose sugar remain attached to one another. So in between you have a phosphate group, and there is the 3' OH is bonded to the one of the phosphate group, and the other phosphate group is bound to the 5' OH group. 5' OH group of the other deoxyribose sugar, and a phosphodiester bond is formed. So the phosphodiester bond is present between it is the bond which is present in your sugar phosphate backbone. So a nucleus does? Nucleus will basically digest the phosphodiester bond. Now there are two ways by which the phosphodiester bond can be digested. Either the phosphodiester bond can be broken from the end. So if the phosphodiester bond is broken towards the end, then it is an exonuclease. Okay. So towards the end, if my enzyme is cleaving the phosphodiester bonds, then it is called an exonuclease. And if the enzyme is cleaving the phosphodiester bond in the middle of the DNA molecule, then it is an endonuclease. Okay, so here you can see in this figure. So this is the figure which has been taken from the textbook T.A. Brown. What is shown over here is that this is an exonuclease, and what an exonuclease does, it removes nucleotide one at a time from the end of a DNA molecule. So these are the ends of a DNA molecule, 5 prime to 3 prime, and the lower DNA strand will have polarity 3 prime to 5 prime. So from the end, you can see from the end of the DNA molecule, your nucleotides are getting removed. The nucleotides are getting removed. And how are they getting removed? Due to the action of this enzyme exonuclease, which is breaking the phosphodiester bond. So your exonuclease is breaking the phosphodiester bond towards the end of the DNA, and so these nucleotides are getting removed. Now, endonuclease, endo means inner, isn't it? Exo and endo, you can easily understand that exo means outer and endo means inner. So if this enzyme is acting in the towards the inner side of the or within a DNA molecule. So your enzyme is acting within a DNA molecule. Then what will happen? So, for example, this is your DNA molecule, and this is the site where your endonuclease is acting. So, the cleavage is taking place from the middle or from within the DNA molecule. The phosphodiester bond is getting broken, and as a result, you are getting different fragments of DNA. So, you are getting different sizes of these DNA fragments. Okay. So it is not cleaving the nucleotides or cleaving the phosphodiester bond which are present towards the end, but it is cleaving the phosphodiester bonds which are present towards the inside of the DNA molecule. So it can cut over here or over here. So as a result, what you are getting different fragments of the DNA molecule. Okay. Now exonucleases can also be. So there can be two ways by this exonuclease acts. Okay, so the two enzymes differ in the number of strands that are degraded when a double-stranded DNA molecule is attacked. So these are the two examples of exonuclease. One is BAL31 and another is exonuclease 3. So if you see this figure, you can understand this BAL31 is acting on both the ends of the DNA. So this is one end of the DNA, this is the other end of the, it is acting on both the ends of the DNA, and it is causing or it is removing the nucleotides from both the ends of the DNA. While exonuclease, what it is doing, it is acting only on the 3 prime end of the DNA. So this is your 3 prime end of the DNA. So it is only cleaving the phosphodiester bond towards the 3 prime end. So your 5 prime end is remaining intact. But this BAL31, what it is doing, it is cleaving at both 5 prime end and 3 prime end. Okay. 5 prime. So the BAL31 removes nucleotides from both strands of a DNA double-stranded molecule, and the exonuclease 3 removes nucleotide only from the 3 prime terminus. Okay, so there can be different types of exonuclease. So why are these different enzymes important? So if you want the 5 prime end to have overhangs, or you want to create a sticky end, so from a blunt end DNA molecule. So from a blunt end DNA molecule, if you want to have a sticky end. Okay. So what is blunt cut, sticky cut, we will understand in the subsequent slide. Then we can use this exonuclease 3. So because once you use the exonuclease 3, then you are seeing that you are having this 5 prime overhangs. Then this 5 prime overhang, DNA molecule having this 5 prime overhang can be used for ligating with the vector which is having 3 prime overhang. Okay. But basically, what is important in this slide is that there can be different types of exonucleases. One type of exonucleases can act on both the DNA strands, and there can be some exonucleases which will act only on one of the DNA strand, or it will act only on one end of the or it is one is acting only on the 3 prime end of the DNA, and another is acting on both 5 prime and 3 prime end. Okay. Now there can be different types of endonucleases as well. So one type of endonuclease is the S1 endonuclease. So this S1 endonuclease is isolated from the fungus Aspergillus oryzae. So this is a fungus, okay, and it cleaves only single-stranded DNA, including single-stranded nicks in many double-stranded molecule. So S1 nucleus only acts on single-stranded DNA molecule. So you see this figure here. So this is a single-stranded DNA molecule, and this S1 nucleus will specifically act on the single-stranded DNA molecule uh, and cause breaks in the DNA. Okay. Or it can also act on nick. So if, for example, a nick has been created, for example, your RNA primer, which was there, that has been removed by, say, for example, DNA polymerase 1, then a nick is created. Now, once this nick is created, then in the double-stranded DNA, there is a small region which is having a single-stranded DNA. So now in the single-stranded DNA, this S1 nucleus can act, and it can cleave this single-stranded DNA. Okay. Then DNase1 cleaves both single and double-stranded DNA. So you see this figure here. So it has been shown here that this is a single-stranded DNA molecule and this is a double-stranded DNA molecule. DNase1 can act on both single-stranded DNA molecule as well as double-stranded DNA molecule. Okay, so it is now this is non-specific as it attacks DNA at any internal phosphodiester bond. So it can act anywhere. The phosphodiester bond at any region within the DNA molecule can be broken. As a result, you are getting different fragments of the DNA molecule, single-stranded DNA molecule. Similarly, here also, if this is a double-stranded DNA molecule, then it can act anywhere within the double-stranded DNA molecule, and you will get variable size fragments of this DNA molecule. Okay. Now, what are restriction endonuclease? So this S1 endonuclease, DNase1, these are all non-specific. It can act anywhere on the DNA. While these restriction endonucleases, why are they so popular in molecular biology? Because they are very specific and they recognize specific DNA sequences. Uh, restriction endonucleases have the ability to recognize specific DNA sequences, and those DNA sequences are called recognition sequences. So each restriction enzyme is unique in the sense that it recognizes different DNA sequences. These DNA sequences are called recognition sequences, and within this recognition sequence, there are specific sites at which this restriction endonuclease will cause a cut. So these sites are called the restriction sites. Okay. So for example, GAATTC. So eco eco R1 will specifically cleave between the G and the A nucleotide bases. Okay. So that is why. So now let's learn about restriction enzymes in detail because they are widely used. Okay. So why are restriction enzymes required? Anyone? So restriction enzymes are one of the most important players in molecular biology because if you want to do any type of cloning, then what has to be done? So this is what is done in a cloning. Basically, in cloning, you have a vector molecule and you have a gene of interest. So both this vector, so these are different names actually, but sometimes some people get confused. What is a vector? What is gene of interest? These are DNA molecules only. Okay, so both of these are double-stranded DNA molecule. So vector is that molecule which will carry your gene of interest. So for cloning, what we do is we cleave the vector with a restriction enzyme, or with a set of restriction enzyme, we cleave the gene of interest with the same set of restriction enzyme, and then we ligate the gene of interest with the vector molecule. So for um, cleaving or for causing a cut in the vector molecule, we need restriction enzymes. Okay. So to clone us, so for restriction enzymes, they're mainly used yeah, required for gene cloning. So to clone a single gene which consists of 2 to 3 kb DNA. So you see here this figure. So for example, you have a gene which is 2 to 3 kb in size, and you want to clone this 2 to 3 kb DNA into a plasmid. Okay, the gene has to be cut out of the large DNA molecule. So your gene is only 2 to 3 kb, but this gene is present in your is present on your DNA, and this DNA molecule can be very long. It can be it is usually greater than 80 kb. So you have to cut this gene from this from your genome. Okay, from the genome or from your long DNA molecule. If this if this gene has to be cut, then we need the restriction enzyme. Okay, then the large DNA molecule may have to be broken down to produce small fragments so that it can be carried by the vector. Now, vectors, there are different types of vector. We will learn what are the different types of vector. Now, each vector is capable of carrying only a particular size of the gene of interest. Okay, so if you're using plasmid vector, then it can only carry your gene of interest, which is less than 10 kb. It cannot carry molecules which are greater than 10 kb. So if you want to clone larger gene fragments, then you have to use different types of vectors. For example, lambda Faj vector or episomal vector. So there are different types of vectors which has to be used, and you must it is the size of the insert is very important. Okay, so most plasmid vectors are very inefficient at cloning DNA molecules which are more than 8 kb in length. So the restriction enzymes are performing two functions. First of all, if you want to insert your gene of interest in the vector, your vector has to be cut. So the vector is cut by the restriction enzyme. So for that you will require restriction enzyme, and for isolating your gene of interest from the DNA molecule, for that also we will require restriction enzyme so that from the large DNA molecule we can isolate the 2 to 3 kb length of gene. Okay. Now, let's learn about the discovery of restriction enzyme. So how was this restriction enzyme discovered? So it was found that bacteriophages. So bacteriophages are viruses which infect bacteria. So it was found that there are certain types of bacteriophages which infect the bacteria, but the bacteria do not develop the infection, or the bacteria cell does not lyse. So bacteria has an immunity by which it can prevent the infection by the bacteriophage. So 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 this was the first thing that was seen, that there are certain types of bacteria which have the ability to prevent the growth of certain types of bacteriophages. And some strains of bacteria were shown to be immune to bacteriophage infection. Now, when this was further uh, when researchers try to further understand that how is this possible, then it was found that the restriction occurs because the bacterium produces. So what happens, you can see in this figure. So this is the bacteriophage, and this is your bacteriophage head, and this is the tail. And bacteriophage in the head, it has its genome, so it can be either DNA or RNA. So for example, this is a DNA molecule, and after infecting the bacteria, what the bacteriophage does, it injects its DNA molecule into the bacterium. Now, once this virus has injected its DNA into the bacterium, then this bacteria has certain enzymes inside it which are called the restriction endonucleases, and this restriction endonucleases binds to the viral DNA, and it cleaves this viral DNA. It chops it into pieces, and as a result the phage DNA gets cleaved and is inactivated. Okay, while the bacteria, so the question was that if these restriction endonucleases are present in the bacteria and it is cleaving the viral DNA, why is it not cleaving its own DNA? So it was found that these restriction endonucleases they have they are sequence specific. They can recognize particular sequences, and the sequences that they recognize, some of the residues, some of the nucleotides are methylated in the bacterial DNA. So you see here, this is the bacterial DNA or bacterial genome, and in the bacterial genome, there are certain nucleotides which are methylated. So the sequences, which are the recognition sequences of the restriction endonucleases, this the nucleotides in those recognition sequences are methylated. Mainly the adenine adenine nucleotide, they are methylated. As a result, the restriction enzyme cannot recognize these site, and and the and as a result, this restriction enzymes cannot act on these DNA molecule and cleave it. So that is why the bacterium's DNA is protected from attack, because it carries additional methyl groups that blocks the degradative enzyme action. Okay. So bacterial DNA is not cleaved, so restriction endonucleases cannot bind to the recognition sequences. Why? Because the recognition sequences are methylated. Okay, so all these figures are taken from the book T.A. Brown. So that is how the bacteria can prevent the infection of Paj. Okay, Paj infection. So now we will learn about the restriction methylase system. So this was explained in detail in lecture in the first lecture of this week 4. So you can see here, so this is EcoR1 enzyme. So this EcoR1 enzyme this is the restriction recognition sequence of EcoR1, GAATTC. So this we will learn about the different types of restriction enzyme, but for now, just remember that EcoR1 is an example of type 2 restriction enzyme. Okay, so it is a type 2 restriction enzyme. So type 2 restriction enzyme have an important property, is that the recognition sequences that they identify are palindromic in nature. What is meant by palindromic? So palindrome means if you read a word from one end to the other end, and vice versa. So for example, see this. So So if you read this DNA sequence from the 5' to 3' end, it is GAATTC. And the lower strand, if you read the sequence of the lower strand in the same orientation, in the same 5' to 3' direction, then also it will read as GAATTC. So such type of DNA sequences are called palindromic DNA sequences. And restriction enzymes have this special ability, or they specifically recognize only palindromic DNA sequences, and this is the restriction site of this enzyme EcoR1. So it cleaves between the G and A nucleotide bases. And so EcoR1 restriction enzyme cuts the DNA. So you get a 5' G and 3' CTTAA. Okay. But this EcoR1 enzyme, so if this sequence is now present in your bacteria, then what happens, that specifically at this adenine nucleotides, there is a methyl group. So this adenine is modified. It has a methyl group in its base, on its base. So because of the methyl group, this EcoR1 restriction enzyme can no longer recognize its own recognition sequence, and as a result, it does not cleave the bacterial's own DNA is not cleaved, while any foreign DNA which is lacking this methyl group, they are cleaved. Okay, so the restriction enzyme recognize non-methylated double-stranded DNA, so it will specifically recognize non-methylated double-stranded DNA, okay, and cut it at specific recognition sites. So this is the recognition site. For example, for the recognition sequence is what GAATTC, while the recognition site or the restriction site is between the G and A nucleotide. So the modification enzymes are paired with restriction enzymes and recognize the same sequence. So modification, so we will learn when we will learn about the different types of the restriction enzyme, we will see that the restriction enzymes have a type of modification enzyme. So there is one subunit which can so there are DNA methyl transferases basically. So these are DNA modifying enzyme. So with the help of this enzyme, the methyl group is transferred onto the adenine nucleotide, and the restriction enzymes are paired with this modification enzyme, so that modification enzymes, what they do, modification enzymes methylate the recognition sequence, which prevent the restriction enzyme from cutting it. So ECOR1, the restriction enzyme cuts the DNA, while the ECOR1 modification enzyme. So the restriction enzyme is paired with the modification enzyme. And so the modification enzyme, once it adds a methyl group to the adenine nucleotide, then the restriction enzyme can no longer leave the leave at the recognition site. Okay. Now, what are the nomenclature of restriction enzymes? You all have learned about the nomenclature, isn't it? So it is EcoR1. If it is EcoR1, the restriction enzyme is EcoR1. Then E stands for your genus, isn't it? The genus of the bacteria. Okay. The CO stands for the species. R stands for what? It is the strain of the bacterium, and 1 stands for the. So it is the order of discovery of the restriction enzyme. So this in this R strain, this was the first restriction enzyme to be discovered. So it is EcoR1. Or the number of restriction enzymes, not inside, not actually the number of restriction. It is the it is something to do with the discovery of the restriction enzyme. So this was the first restriction enzyme from E. coli R strain to be discussed. So it is EcoR1. Okay. So the first letter is your genus name. Genus name is capitalized. Next two letter is the species name. The strain is then this letter comes from the strain of the bacteria. So R in EcoR1 refers to the E. coli strain RY13. Okay. So from the RY13 E. coli strain, this enzyme has. So the R stands from comes from this strain numbered RY13. Okay. Now, there are different types of restriction enzyme. So there are basically three types of restriction enzyme as I was discussing. One is restriction enzyme type 1, restriction enzyme type 2, and restriction enzyme type 3. Now, out of all these restriction enzymes, the restriction, the type of restriction enzyme which is important to molecular biologists are the type 2 restriction enzyme. Okay, so this type 2 restriction enzymes are most widely used and in molecular biology. So why is it so? So we will understand once we discuss all the different types of restriction enzymes. So what does type 1 restriction enzyme do? So first of all, first is that this type 1 restriction enzyme cut the DNA 1,000 or more base pair away from the recognition site. Okay, so it has a particular recognition site, but the recognition site and the restriction site are different. Okay, so the DNA sequence which it is recognizing is different, and the DNA site at which it is cleaving that at which it is cutting the DNA, that is 1,000 base pairs away from this recognition site. So the recognition site is that site where at the site at which your restriction enzyme will come and bind to that particular DNA sequence. And this site, the site where it binds to the DNA, it does not cut at that site, but it cuts the DNA 1,000 base pairs away from its recognition site. Okay. So how does it do so? So you can see in this figure. So this particular DNA region which it recognizes is has been marked in green. So this is the recognition site, but it is cleaving the DNA which is 1,000 base pairs away from this. So how does it do so? So for that, it what it does, it causes the DNA, or it creates the DNA to form a loop. So once this restriction enzyme comes and binds at this recognition site, it creates a loop in the DNA, so that the DNA base sequence, which is 1,000 base pair away from this recognition site, that can come to its close proximity. So once it has come to its close proximity, then it can cleave that particular DNA strand. So you see here that this restriction enzyme 1, type 1 restriction enzyme, it has three different subunits. So it is a single enzyme, but it has three different subunits. So one is HSDS. So HSDS is the subunit which recognizes. So this is the specificity subunit. So this is the subunit which will bind to or which will recognize that particular DNA sequence. Then another is HSDM. M is the modification subunit. So this subunit of restriction enzyme, it will catalyze the transfer of methyl group onto the DNA nucleotide. Okay, and this HSDR, this is the restriction subunit. So restriction subunit, what will it do? It will cleave the DNA strand 1000 base pair away from the recognition site. Okay, so why is the DNA? It is necessary to cause a loop in the DNA because its recognition site is or its restriction site is 1000 base pairs away from the recognition site. So what it does, it loops the DNA around so that the enzyme binds both at the recognition site and the cutting site. So it is binding at the recognition site and at the same time it is also binding to the cutting site by causing a loop in the DNA. Now the type 1 restriction system consists of a single protein with three different subunits. So this is a single protein, but it has three different subunits, HSDS, M, and R. So the specificity subunit, what it does, it recognizes the DNA. Modification subunit will methylate the recognition sequence, and the restriction subunit cuts the DNA at a distance from the recognition sequence. Okay. Since the exact length of the loop is not constant, and since the base sequence at the cut site is not fixed, these enzymes are of little practical use to molecular biologists. Okay, so now this 1000 base pair away, this is very vague. Sometimes it can cut 98 base pairs away. Sometimes it can cut, for example, 1100 base pair away. So this is not fixed. And if we are using this restriction enzyme, then we will, if we want to cut at a particular site, then that cannot be regulated. So since it is not fixed, so these type of restriction enzymes are not used in molecular biology. Okay. Because the cut site is not fixed. Now, the second type of restriction enzyme, there are type 2 restriction enzymes. These are the restriction enzymes that are mainly used. And so one of the main feature of restriction enzyme type 2 is that it recognizes the palindromic sequences. So palindromic sequences read the same from the 5 prime to 3 prime and in the reverse 5 prime to 3 prime in both the strands. So if you read, so this is your upper strand, this is your lower strand in the 5 prime to 3 prime direction. This is reading the same as it is reading in the lower strand in the 5 prime to 3 prime direction. So they are the recognition sites for type 2 restriction enzymes and create symmetrical cutting patterns that generate complementary sticky ends essential for molecular biology or molecular cloning. Okay, so in the 5 prime to 3 prime strand, if it is cutting between the G and the A nucleotide, then in the lower strand also it will cut between the G and the A nucleotide. Okay, so what you are left with, so once it causes this curve, you are getting a 5 prime overhang, isn't it? So this is 5 prime overhang. So one of these strands will have this AATTC and this short G and this other strand. Now these are actually called the sticky ends. These are called the sticky ends. Why? Because of the natural tendency of the A nucleotide to base pair with T. So if these two molecules, this strands, once it is cleaved, but because this A nucleotide has the tendency to base pair with T, these will again come and base pair with each other by forming the hydrogen bonds. That is why they are called the sticky end, because they are always trying to come together because of this hydrogen bonds between the complementary base pairs. But so if you see this figure, then this HPA1 and EcoR1, both of these restriction enzymes are having the same recognition sequences. Both of these are recognizing this GTTAAC DNA sequence, but one of this restriction enzyme is causing a sticky end, or it is causing a staggered cut. This is called a staggered cut, and another one is causing a blunt cut. So it is cutting the DNA from the center of the DNA molecule. Okay, symmetrically it is cutting the DNA molecule, but the other one EcoR1, what it is asymmetrically cutting between the G and A base pair. Okay, and the A nucleotide bases. So that is why this HPA1 is causing blunt ends, while this EcoR1 is creating sticky ends. Okay. So now the type 2 restriction enzymes cut DNA within the recognition sequence. Since the exact position of the cut is known, these restriction enzymes are typically used in genetic engineering because we know exactly. So there are different types of restriction enzymes, and you can find a list in any standard molecular biology book. You will find that there is a list of restriction enzyme and what is their recognition sequence. So there will be a table where you will find that these are the different type of restriction enzymes and these are their recognition sequences. Okay. Now, some will what will do now this type 2 restriction enzymes, they can do both. They can either create blunt end or they can create sticky ends. Okay. Now, sticky ends are more convenient than blunt end when joining together fragments of DNA using DNA ligase. Now, mainly what is done is if we have, for example, in our lab, we have these both of these enzymes. We have HPA1 and we have EcoR1. Both of these are present in our lab. And now I want that I want to do a cloning. Uh, so what will I do? I will not use this HPA1, but rather I will use this EcoR1. Why? Because this EcoR1 can produces sticky ends, and these sticky ends are easy to ligate. The chances of forming or of cloning my gene of interest into the vector is higher if we are using restriction enzymes that cause sticky ends. And why is it so? Because because of this tendency of this complementary base pairing. Okay. So if your vector will also have the complementary basis, then your vector and your gene of interest, they will come together, and then by using DNA ligase we can ligate the two DNA molecule. Okay. Now in type 2 restriction systems, one more point to remember is that the modification enzyme and the restriction endonuclease are completely separate proteins. Okay, they are separate proteins. As I was mentioning that there is a different enzyme which is DNA DAM methylase. Okay, or DNA methyl transferase, which causes the which adds the methyl group to the adenine nucleotide. And while as we were seeing in type 1 restriction enzyme, that there is this one protein, and this protein is having three different subunits, and each of the subunit is having a different function. One is recognizing the sequence, one is modifying the DNA, and another is cutting the DNA at that restriction site. But here in type 2 restriction enzyme, the modification enzyme is separate from the restriction. So the modification protein is different and the restriction protein is different. Okay. Now, now they will come across this term frequently. What is the start activity of restriction enzyme? So start activity basically means that if you are doing any cloning experiment and you have set up a digestion reaction, okay, restriction digestion reaction. So what is done in the laboratory basically is if you want to clone a gene of interest into a vector, first of all, you will check in the lab that first of all, what is required if you want to clone a gene of interest into a vector? You know the gene sequence. Okay, you know the gene sequence. Now what might happen is that the end of your gene of interest might not have the DNA sequence which is recognized your uh, uh, by the restriction enzyme that you have in your laboratory. Okay, so for example, you have BAM-H1 in your laboratory. Now, this BAM-H1 will recognize a particular recognition sequence. It will recognize a particular DNA sequence. Now, you might not have that DNA sequence in your gene of interest. Then what you can do? We can design primers. So what is done? We will design primer. So we will design primer such that the primer will have the recognition or will have the DNA sequence of BAM each one. Okay. Then what we will do? We will do a PCR reaction. So in the PCR reaction, what we will get? We will get the gene of interest having this BAM H1 site. Okay, BAM H1 site. Similarly, we can design a reverse primer with, for example, SAL1. You have SAL1 in your laboratory, but this SAL1 is not present in your gene of interest. Then what we can do is we can synthesize a reverse primer which is having the SAL1 recognition sequence. Then in the DNA that will get amplified. The DNA sequence that will get amplified will have your SAL1 recognition sequence as well. Now, what we do is that we take this amplified DNA product, right? And then so first we will run a gel. We will check if the amplification has taken place. We will analyze it on an agarose gel by running an agarose gel electrophoresis. And then what we do, we take this amplified product and then we set up a small reaction where we add both these restriction enzyme, BAM H1, SAL1, we add the buffer and then we incubate it at particular temperature. Okay. Then what will happen? We will get then what we once this amplified product is treated with BAM H1 and SAL1, you will get the sticky end. So one of your DNA strand will have this BAM H1 uh, uh, BAM H1 recognition sequence, and on the other end, you will have this SAL1 recognition sequence. Now, we'll take the vector also. Now, it will take now if your vector in the multiple cloning site of your vector, you will have this BAM H1 and SAL1 site. So you will digest the vector with BAM H1 and SAL1, and then you will do a ligation. Okay, you will ligate your gene of interest with your vector, and I will set up a ligation reaction. And then you will, once you have the ligated product, you will again run a agarose gel electrophoresis, and you will again do PCR and restriction digestion to check yes if your gene of interest has been cloned in your vector, and then you will transform it in the bacteria. So what happens sometimes that if you have very little the DNA template that you're taking, the DNA template that you're taking is in very less quantity compared to your restriction enzyme. So if you have taken the restriction enzyme in a very high amount, then what will happen? The restriction, and if you have incubated it for a very long time. Okay, for a very long time, if you have incubated the restriction enzyme with your DNA template, then the restriction enzyme can go and cleave at anywhere in the DNA sequence. It will cleave anywhere between the in the DNA sequence. So once it has cleaved at particular site, then and it is not finding any recognition sequence to cause the cut, then it can cleave anywhere in the DNA sequence, and your DNA will get uh, you will not have your required DNA sequence with having those restriction sites. Okay, so star activity occurs when there is excess enzyme for the amount of DNA. So compared to your DNA template, if you have added excess amount of restriction enzyme, then your restriction enzyme will, what will it do? It will cleave at the nucleotide sequences that differ slightly from the exact recognition sequence. Okay. Exact recognition sequence. So, for example, it's a GAATTC, but it can cleave after cleaving that particular DNA sequences. And if you have incubated it for a very long time, it is not finding where it will cleave, then it can cut anywhere or anywhere in the DNA sequence. So, the activities are laboratory artifacts, and most restriction enzymes are sold with a specific. Now, so nowadays, this star activity is usually not found because there are specific buffers that come with this restriction enzyme. And as long as this buffer is used with a sufficient amount of DNA, then the star activity can be avoided in the laboratory. Okay. So remember the star activity, what is it? So the restriction enzyme may cut DNA at nucleotide sequence that differs slightly from the exact recognition sequence. And when can it happen? It can happen if you have added an excess amount of restriction enzyme compared to your the DNA that you want to digest, or if you have incubated it for a very long time. Okay. Now this is the table which was there in the video lectures of week 4. You can see that what are the differences between the three types of restriction enzymes. So first difference, main difference is what? That only type 2 restriction enzyme can recognize palindromic sequences. So the recognition sequences of type 2 restriction enzymes are palindromic, while for type 1 and type 3, it is non-palindromic. Now, type 1, another disadvantage is that it is not cleaving at its recognition site. It is cleaving 1000 base pairs away from the recognition site. So this is one of the disadvantage of type 1. And for type 3, also the disadvantage is same, that it is not fixed. It is cut. It can cut anywhere, which is 20 to 25 base pair away from the recognition site. But type 2 restriction enzyme always cuts within the recognition sequence. Now enzyme composition, as we learned that type 1 has three different subunits. It is one protein with three different subunits: HSD-R, HSD-M, and HSD-S. Now type 2 has three subunits: RMS, and type 3 has RES and MET. Okay. So these things were discussed in detail. I'm not going to detail. So cofactor that is required for the activity of the restriction enzyme, it is magnesium, S-adenosyl methionine, ATP. For type 2, it is magnesium. For type 3, it is ATP. Now, product, it is non-sticking end. Type 2, it is sticky end and blunt end also. And type 3, it is non-sticking end. Okay. Now, the type of recognition sequence does type 2 restriction enzyme have? So what is the type of recognition sequence that is recognized by type 2 restriction enzyme? So just now we learned option A is non-palindromic, option B is palindromic, option C is inverted repeats, and number D is random. So type 2 restriction enzymes recognizes DNA sequences which are palindromic. Okay, so option B is the answer. Okay. So we discussed just now. Now the property that is unique to type 1 restriction enzyme is what? So the type 1 restriction enzyme, I've been explaining this several times. So it recognizes cleavage site, which is 1000 base pair away from the recognition site. For type 3, it is 20 to 25 base pair away. For type 1, it is 1000 base pair away. Now type 1 generates sticky ends. This is false. It generates non-sticky ends. And it cuts within the restriction recognition site. This is also wrong. False. Requires only magnesium. So only magnesium is required by type 2. Type 2 restriction enzyme only requires magnesium as cofactor, but type 1 requires other cofactors also. Magnesium, then it requires S-adenosyl methionine. All these are required. Okay, so you go through this table very carefully. And ATP also. Okay, so answer is option A. Now the composition of type 3 restriction enzyme is what? So already the answer is given. So it is HSD-R. Okay, the composition of type 3 restriction enzyme, what is it? So the recognition site has two separate non-palindromic sequence arranged inversely orientated. The cutting site is 20 to 30 base pair away from the recognition site, and the type 3 enzyme is composed of two subunits. One is RES and another is MOD. So these are few things that you need to remember. These are there is no not you do not need to understand much, so you just need to remember that what are the subunits which are present in type 3 enzyme, subunits present in type 2 enzyme, and subunit present in type 1 enzyme. So it is RES and MOD in type 3 enzyme. Okay, you go through the table that is there in the in your lecture slides. Okay, so it is RES and MOD. So the MOD subunit is required for modification of the host DNA, whereas RES subunit, what it does, it cuts the unmethylated DNA. So this covers the first section, that what are the restriction enzymes, different types of restriction enzyme, why are they required, what is the mode of action of different restriction enzyme, which type of restriction enzyme is used mainly for molecular cloning. So all this covers the first part. Then we will move to now this is another part. So this is the mode of action. So this is again taken from the lecture slide. So type 2 restriction enzyme, how do they act? So what is the mode of action of type 2 restriction enzyme? So you can see here this type 2 restriction enzyme, it has three different proteins, R, S, and M. Okay, R, S, and M. So the type 2 restriction enzyme have specific recognition DNA sequences. They act as specific recognition sequences to rapidly identify the site. What does the enzyme do? The enzyme scans the stretch of DNA through a non-specific interaction and diffusion along the length of the DNA. To expedite the recognition process, enzyme does not make interaction with the bases. Instead, it makes contact with the DNA backbone. So this restriction enzyme, first what it has to do in the entire DNA molecule is it has to find its recognition sequence first. So that is one of the important thing, that how is it able to find this recognition sequence in this entire DNA molecule. So how does it do that? So it scans so there is a diffusion model which says that this recognition sequence, rather than forming any base pairs with the nitrogenous bases, what it does, it diffuses along the length of the DNA through non-specific interaction, and it recognizes the so it only makes contact with the DNA backbone. Then what it does, once it has found its recognition site, then the enzyme will make specific interaction with the nucleotides which are present at the recognition site. So the nucleotides which are present at the recognition site, those recognition site will be recognized, and it will bind to these recognition site. Now, at this site, what will happen? Now it will interact with the nucleotide bases by hydrogen bond van der Waals interaction, and a closed complex is formed. So a closed complex is formed between the DNA and its recognition sequence. So the DNA closed complex formation induces major conformational changes in the restriction enzyme, and then after that, the cleavage reaction takes place. So a closed complex is formed between the restriction enzyme and the recognition sequence which is present in the DNA. Once this is done, then it will cause some modification in the DNA molecule, and it will cleave at the particular site, and this your restriction enzyme will be released. So once the closed complex is formed, it will activate the cleavage activity of the enzyme, and it will result in the introduction of DNA break on both strands to give fragment with 5' phosphate group and 3' OH group. Now this restriction methylase system, this is again from the lecture slide. So what does it say? That how this restriction enzyme is able to distinguish between self and foreign DNA. So one of the concept is that the precise mechanism of this distinction is not known. Okay, but there are some studies which what do they suggest? They suggest that in the absence of methylation, a closed complex is formed, and it allows that the proper activation of cleavage activity of the enzyme. But the presence of methyl group on nucleotide does not allow formation of closed complex, and consequently enzyme falls from the DNA. Okay, so just now we learned that once this restriction enzyme has recognized its target DNA sequence, it will bind to that and it will form a closed complex. Now, once this closed complex is formed between the DNA molecule and the recognition and your restriction enzyme, then it will cause some conformational change in that recognition sequence, and the catalase activity, or the catalytic activity will be activated. Now, once the catalytic activity is activated, it will cut the DNA molecule. But what happens if the DNA is methylated at some nucleotide residues? For example, it is methylated at the adenine nucleotide residues. Then this restriction enzyme can no longer form a closed complex. Now, if the closed complex is not formed, then the catalytic activity will also not be activated, and there will be no cleavage activity of the restriction enzyme. It will not cut the DNA. Okay. So this is one of the question. that comes directly if you have understood the concept. So in the restriction methylase system, the formation of closed complex is prevented by. So how is the formation of closed complex prevented? Just now we learned option A is presence of a methyl group, option B absence of magnesium ions, option C is presence of ATP, and option D is absence of SAM. So the closed complex was not able to form. Why? Because of the presence of the methyl group. Just now we learned in the previous slide only. Now, if there is a methyl group, then the closed complex is not formed, and as a result, the the catalytic activity of the restriction enzyme will not be activated, and the DNA will not be cleaved. Okay, so answer is option A. Now, what is the function of DNA ligase? So after you have created the digested their DNA of interest. So as I was explaining that what are the different steps. So if you have done cloning anywhere in laboratory, you might be knowing that first you need to know the DNA sequence. Now, once the DNA sequence is known, and if you have your recognition sequence present in the DNA, then first of all, so if you do not have the recognition sequence present in your gene of interest, you need to amplify it with forward. You need to design the forward primer and reverse primer carrying the recognition sequences that you have in your laboratory. So if you have a particular set of restriction enzyme which is present in your vector, and it is also present in your, and you want that gene of interest to be cloned in that vector which between those two particular sites, then what you will have to do, you have to take your gene of interest and you have to amplify it. You have to do a PCR reaction. You have to do a PCR reaction by using forward primer and reverse primer. The forward primer and reverse primer must have the recognition sequence, must carry the recognition sequence of the restriction enzyme that you have in your laboratory. Okay, having the recognition sequence, once this DNA is amplified, then what we do, we set up a digestion restriction digestion reaction. So once you set up the restriction digestion reaction, then you will have your gene of interest with the sticky ends. Okay, now you always, what you will do, you will also set up the digestion reaction of the vector. Okay, so you will set up the digestion reaction of the vector. So this was the digestion reaction of your amplified DNA product. Amplified DNA product. Okay, there's a digestion reaction of the, you will set up the digestion reaction of the vector, and after that, once you have the digested vector and the digested gene of interest, then you will do a ligation reaction. So for ligation reaction, what is required? You require DNA ligase. Okay, and one of the most commonly used ligase in laboratories is the T4 DNA ligase. Okay, not bacterial DNA ligase, it is T4 DNA ligase which is used. So what does ligase do? So you see this figure here. So the main function of ligase is that it will seal the phosphodiester bond. So for sealing the phosphodiester bond, what is required? It requires a 3' OH group and a free 5' phosphate group. So once it will find that it has a 3' free OH group and a 5' phosphate group, then it will bind to those region, and with the help of, by taking the energy obtained by hydrolysis of ATP, it will seal these two ends. It will seal the two ends of the DNA. Okay. So DNA ligases are molecular glues basically. So they will link segments of DNA together permanently. Now ligating blunt end is very slow. So that is what I was explaining. So what is usually preferred? Usually enzymes, if you are doing any molecular cloning, then usually restriction enzymes that create sticky ends are usually preferred rather than enzymes which cause blunt end. Why is it so? Because it is very difficult to ligate DNA molecules which are having blunt ends. Okay, because it is very slow and requires a high concentration of DNA ligase as well as high concentration of DNA. So, for ligation of DNA which are having blunt end, what will be required? Because now because they are having blunt ends, this the DNA of the vector and the DNA of the gene of interest will not show any kind of binding interaction. There will be no binding interaction. Like if you have sticky ends, then the sticky end of your vector and the sticky end of the gene of interest, they will show some binding interaction with the help of the hydrogen bonds. There will be complementary base pairing, and the two DNA molecules will come together. But for if you want to stick DNAs which are having blunt end, then what will happen? You need a very high concentration of your insert, because if you increase the concentration of your insert, then only the insert and the vector molecule will come together, or there will be high chances that probability that your DNA molecule gene of interest will be in close contact to your vector. Okay, so that is why you require a very high concentration of the DNA. Okay, so the reason is that now, another thing is that bacterial ligase cannot join blunt ends at all. Okay, we do not use bacterial ligase in labs. Okay, T4 ligase is mainly used. So T4 ligase is the ligase which is obtained from the T4 bacteriophage. And why is ligase used? Because T4 ligase can ligate both blunt ends and sticky ends very efficiently. So the ligation of complementary sticky ends is much more efficient. Why is it so? This is because compatible sticky ends can base pair with one another by hydrogen bonding, forming a relatively stable structure for the enzyme to work on. So that is what I was explaining. So if they are having already they are having sticky ends, then the two ends, the DNA which are complementary to each other, they will come together because of this hydrogen bonds. And because they come together, this ligase can work more efficiently. But this is not the case when we are when we are trying to clone a DNA having blunt end with our vector. Okay, so although T4 ligase hydrolyzes ATP as a source of energy, bacterial ligase uses nicotinamide adenine dinucleotide, which is splits into AMP plus nicotinamide mononucleotide. So this there is a figure that was explained very nicely in the lecture. So you can go through that figure. So always remember that T4 ligase uses ATP. So it uses the energy that is obtained from hydrolysis of ATP to seal the phosphodies to create to seal the 5' phosphate group with the 3' OH group. Okay, the DNA strand which is having 5' phosphate group with the DNA strand which is having 3' OH group. Okay, but the DNA bacterial ligase do not use ATP, rather it uses NAD. It uses NAD, and NAD is split into AMP plus NMN, and this energy is used for ligation. Mhm. Now, T4 DNA ligase recognizes the break in the DNA backbone and uses energy from the hydrolysis of ATP to drive the ligation reaction. So, the DNA ligase in bacteria, why is it required? So, it is not only used in molecular cloning. The bacteria has its own DNA ligase. Why does it require DNA ligase? So, if you have read the process of DNA replication, you must be knowing that when these Okazaki fragments are created during polymerization, so these different short DNA strands needs to be ligated. So for that also you need DNA ligase. Then, for example, in your also when the for this template strand, you have a template strand which gets replicated. So the first there is an RNA primer which has to be attached first, and then this RNA primer is extended by the DNA polymerase 3, isn't it? So that when this RNA primer is removed by DNA polymerase 1, then also you get a nick, or yeah, a nick is formed. So those broken ends needs to be ligated. So which enzyme does all this in the cell? It is the DNA ligase. Okay. And we are using now this DNA ligase for molecular cloning also. We're using this structure. Now there is another enzyme which is alkaline phosphatase. Phosphatase means what? Nucleus. What does it mean? It was degrading your nucleic acid. Phosphatase means what it will do? It will degrade your phosphate group. Okay. So basically you see this figure. So see this figure. So your DNA will have 5'. This will be a 5' to end, and this will be the 3' end of the upper strand. and here there will be 3 prime to 5 prime end. So always what has a DNA always has a 5 prime phosphate group and a 3 prime OH group. Now if you add alkaline phosphatase to the DNA, what an alkaline phosphatase does, it will cleave this phosphate group. Okay, it will cleave this phosphate group which is present at the 5 prime end, and it will add an OH group. Okay, now why is this alkaline phosphate is very important in cloning? Because what happens is when you digest your vector, okay, so you have used a restriction enzyme which is causing sticky ends or staggered cut. Then what will happen? It has created the break, or it will convert this double-stranded vector molecule into a single-stranded DNA molecule. But because these are sticky ends and they have the affinity to come together by forming hydrogen bonds, they can self-ligate. So before rather than ligating with the vector, or rather than ligating with your gene of interest, what it will do, it will rather come together and it will self-ligate. Okay, so you see here this figure in that was explained in the lectures. So you can see that this is the P 5 prime P and this is the 3 prime OH. So you have cleaved it by the action of restriction enzyme. But what has happened is, because it has this free 5 prime phosphate and the free 3 prime OH, it can self-ligate. Or if it has sticky ends, then also it will self-ligate, and you will not get your gene of interest cloned into the vector. So to avoid this, what is done? Alkaline phosphatase is added. So what this alkaline phosphatase will do? It will cleave, it will remove this phosphate group and it will add an OH group. Now, like ligase, this DNA ligase will not act if it has a 5 prime OH group and a 3 prime OH group. So it cannot ligate the two ends. So your self ligation is prevented. Okay, self ligation. Self ligation means what? Self ligation means your vector is ligating with itself only. It is not ligating with your gene of interest. Okay, but your what this is your gene of interest. Gene of interest has your gene of interest will have your 5 prime phosphate group and your 3 prime OH group. So now, when you will add the ligase, what will happen? There will be no self-ligation, but this vector will ligate with your gene of interest, because your gene of interest has your 5 prime phosphate group and there is a 3 prime OH group which is present in the vector. So 3 prime OH, 5 prime phosphate, there will be ligation. And similarly, there will be 3 prime OH in this gene of interest and 5 prime phosphate in your vector. Okay. Or basically what happens, so this is one thing to note, that this is 3 prime OH and this will also be 3 prime OH. Okay, so how this 3 prime, this is 5 prime phosphate, this 5 prime phosphate and this 3 prime OH will come together, and your ligase will act on it and it will seal the end. Similarly here also, but here what you will have this OH group which is present in the insert, and this 3 prime OH is also present on your vector. So this OH and OH, how will they ligate? Do you understand what I'm trying to say? So if it is phosphate here and you will have OH here, then ligation can take place. This ligase will act and it can ligate the one end having 5 prime phosphate and the other having 3 prime OH. But there will be one end where it will have 3 prime OH which is present in your gene of interest, and your vector will also have this OH group. Okay, because you have treated the vector with alkaline phosphate. Now all the phosphate have been converted into OH. So all this N 5 prime, 3 prime, 3 prime, 5 prime, all these have OH, 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 OH. Okay, so how will it ligate? So what happens basically is it is not able to ligate. So there are nicks. It is not ligated. There will be broken ends. But when this is transformed into the bacteria has its own DNA repair machinery, and using its repair machinery, it will ligate the two ends. This three OH which is present in our insert and OH which is present in our vector, that will be ligated by using the DNA repair machinery of the bacteria. So these are very important concepts when you are doing molecular cloning. Okay, so the role of DNA ligase in molecular cloning. Now answer this question if you have understood the concept. What it is doing, DNA ligase, cutting DNA, amplifying DNA, joining two DNA strands, removing terminal phosphate groups. Now cutting DNA, who does cutting? It is restriction endonucleases cuts the DNA, or any type of nucleases will cut the DNA. Who amplifies the DNA? Polymerases. So this option is wrong. Joining two DNA strands. This is ligase. This is done by ligase, isn't it? So option C is the correct answer. And which enzyme removes terminal phosphate groups? Just now we learnt about alkaline phosphatase. Alkaline phosphatase. Okay, so this option is also wrong. So DNA ligase option is C. Okay. Now linker. Now this concept was also discussed. What is a linker and what is an adapter? Now, for example, you are doing a blunt-ended. You have digested your gene of interest with a restriction enzyme which causes blunt end. You do not have a restriction enzyme which can create sticky end. So in that case, what can you do? So in that case, what is done is that a linker molecule is used. Linker molecule is basically a oligonucleotide. Okay, it is a oligonucleotide which will have your recognition sequence. Now, these things are not done nowadays in the lab. Okay, in the lab, this linker and adapter, they are not used frequently. Rather, what I was explaining previously, so you design forward primer and reverse primer having your recognition sequence, and then you amplify your gene of interest, and then you digest your amplified product with your restriction enzyme. That is followed in the laboratory, but some cases you can use linker as well. Okay. So what is it? It is an oligonucleotide which will have your recognition sequence. Okay. Okay. So if you have a for example, you have a gene of interest. You have a gene of interest, but your gene of interest does not have a recognition sequence, which can be cleaved by the restriction enzyme that you have in your lab. Then how will you clone your gene of interest? So in that case, you can use a linker. Okay. So you see, this is the linker molecule. So this is your entire linker molecule. This entire thing is your linker molecule. So your linker molecule is having a particular recognition site, recognition sequence. For example, this linker, which is shown in this figure, it is having a BAM-H1 site, and you have your blunt-ended DNA molecule. Now, what you do, you will set up a ligation reaction where you will have this blunt-ended DNA molecule, and you will add the linker molecule in the reaction as well, and you will add the ligase. So once you add the DNA ligase, what will happen? These linker molecules will be ligated with your blunt-ended DNA molecule. Now, after this linker molecule has been attached to your DNA molecule, then you can treat it with BAM-H1. So once you treat this DNA molecule which has been ligated with the linker molecule and you treat it with BAM-H1, then you will get these linkers will be cleaved, right? This BAM-H1, these all will be cleaved, where they will be cleaved. And also this one, this main molecule, your gene of interest, which you want to clone in the vector. So your vector also has this BAM-H1 site. So you want to clone this gene of interest having this BAM-H1 into the vector which is having BAM-H1. So this is your gene of interest. Now your gene of interest, you have converted the blunt-ended molecule to sticky-ended DNA molecule. Now you can use this gene of interest to set up the ligation reaction. Is it clear? So from a blunt-ended molecule, if you want to get a DNA molecule which is having sticky ends, then you can go for using the linker molecule. Is it clear? So this figure, if you look at this figure and see the steps, you will understand. No need to go through all these text. Okay, you can read the text for your reference I have added, but if you understand the figure, then you can solve the questions also. You will understand the concept. Okay, so the linkers are short pieces of double-stranded DNA of known nucleotide sequence. So they are oligonucleotides, basically double-stranded DNA molecule, and the sequence is known. And the typical linker is blunt-ended but contains a restriction site. So linker molecule, you see there are no sticky ends in your linker molecule. It is also blunt-ended. and it will have a restriction site. For example, in the figure here, there is a BAM-H1 restriction site. Now the DNA and ligase, what will it do? It will attach linkers. So first you set up the ligation reaction. I was mentioning the ligase will attach linkers to the ends of the large blunt-ended DNA molecule. Although a blunt-ended ligation, this particular reaction can be performed very efficiently because synthetic oligonucleotides such as linkers can be made in very large amounts and added into the ligation mixture at a high concentration. So for blunt-ended ligation, you require very high concentration of your linker molecule, and they are commercially you can purchase them, so you can add them in a very high amount to your DNA molecule, and you can set up the you can add the DNA ligase. Your ligase will ligate the linker molecule with your blunt ended molecule. Now what will happen once you set up the ligation, there will be more than one linker which will attach to the end of the DNA molecule and will produce the chain structure. Now digestion with BAM-H1 cleaves the now you digest with BAM-H1. Once you digest with BAM-H1, it will cleave the chain at recognition sequences, producing a large number of cleaved linker and the original DNA fragment now carrying BAM-H1 sequence. So now your original DNA fragment, initially it was not having a BAM-H1 site, but once you went through this entire process, you created the DNA molecule having this BAM-H1 site. So this modified fragment is ready for ligation into a cloning vector restricted with BAM-H1. So you also need to digest your vector with BAM-H1. Okay, so is this point clear? Now, other than linker, so answer this question now if it is clear. So the role of linker molecule in molecular cloning is what? Amplify DNA? No. Remove terminal phosphate groups, cut DNA at specific site, and option D is provide cohesive ends. So what is its function? It is providing sticky ends. Cohesive ends means sticky ends only. Okay, no need to get confused. So your linker molecule is providing your DNA molecule with sticky ends or cohesive ends. Answer is option D. Okay, you can, the explanation is there in the previous slide. You can go through, that is the entire explanation for this question. Okay. Now adapter is also similar to linkers only, but linker molecules are having they're blunt-ended DNA molecules, but adapter molecules are have sticky ends. Have sticky ends. Okay, so you see this is an adapter molecule which is having a BAM-H1 sticky end. Now, if you so this is a typical adapter. So all these figures are taken from the book T.A. Brown. You can go through that book. So these concepts have been explained very nicely. So the adapter could ligate to one another. So if you have sticky ends, then what will happen? Your adapter molecule, just your adapter molecule. Now you do not have one molecule of adapter. When you are taking, for example, 5 microgram adapter or 10 microgram of adapter, there will be several molecules of adapter. Now one molecule of adapter can ligate with another molecule of adapter, right? That can happen, could ligate to one another because this adapter has sticky ends, so they can ligate with one another, and a blunt-ended molecule will be produced. So then, what is the purpose of using adapter having sticky ends? Okay, now so this can happen. So the new DNA molecule is still blunt-ended. So even you are using adapters, in these adapters you are having these sticky ends, but still these adapter molecules are ligating with one another, and you are getting a blunt-ended molecule. So then the purpose of using adapter is it is not solving the purpose, isn't it? This was blunt-ended and now you are getting blunt-ended again. Once if you ligate, if you digest it with BAM-H1, again it can come and ligate with the other adapter molecules. So how is this then? How is it prevented? How is this ligation prevented? Ligation of one adapter molecule with other adapter molecule. So what is done basically is this. This is 5' end, isn't it? So this will have a phosphate group. This is 3' OH end. Similarly, for this, you will have a 3' OH end. Okay, and you will have a 5' phosphate end. So basically, this adapter molecule is treated with what? What do you have to treat it with? Just now I explained that you have to use alkaline phosphatase (AP). So once you treat it with alkaline phosphatase, then your adapter molecule will not have 5' phosphate group. It will only have OH group. So once it has OH group, then there will be no self ligation. Okay, so the adapter molecules, they are synthesized. They are synthesized in such a way that they do not have 5' phosphate, or they have been synthesized, or they have been already treated with alkaline phosphatase. So the adapter molecule that you get, that you commercially purchase, those adapter molecules do not have 5' phosphate group. They have 5' OH group. Okay, because they have already been treated with alkaline phosphatase. So that solves the purpose, isn't it? So now you will have DNA. So your DNA will be attached to your the adapter, which is having the BAM one side, and then you can ligate it with the vector. Okay, so adapter, like, you can read all these things. Okay, so I explained it through the figure. It is a short oligonucleotide, but it is synthesized so that it already has one sticky end. So unlike your linker, which was having blunt ends, it has already has one sticky end. So the idea is to ligate the blunt end of the adapter to the blunt ends of the DNA fragment to produce a new molecule with sticky end. So the blunt end of the adapter will be ligated to your DNA fragment, and you will have this because your adapter has sticky end, it will have your DNA molecule will have the sticky end. So, but the adapter molecules are synthesized so that the blunt end is the same as the natural DNA, but the sticky end is different. So what that is what I was explaining, that the sticky end is just treated with alkaline phosphate. Because it is treated with alkaline phosphate, is it has the 3' OH is present, but the 5' phosphate is replaced with 5' OH. And as a result, the DNA ligase is unable to form a phosphodiester bridge between 5' OH and 3' OH. Okay, that result is that although base pairing is always occurring between the sticky ends, base pairing will occur, isn't it? Because there is complementarity between the two strands. So base pairing by hydrogen bonds, they will occur, but there will be no ligation because there is a 5' OH and 3' OH. So ligase cannot act. After the adapter has been attached, the abnormal 5' OH terminus is converted. Okay. So that is all about adapter. Now coming to cloning vectors. So what are the different vectors used? What is a cloning vector, first of all? So what is a cloning vector? That it is a molecule. Vector means that it will carry something, right? So cloning vector is a molecule which has the ability to replicate on its own, and it has the ability to carry your gene of interest. So that is your cloning vector. So it can replicate itself inside a cell, and it is used for carrying clone gene or segments of gene in uh, uh, segment of gene to your host cell. Okay, so cloning vector will must have two properties. First, it must be able to replicate on its own, and the second property is that it must be able to carry the gene of interest to a host cell. Now, there are different properties of cloning vectors. The first property is that the vector should be reasonably small and manageable DNA molecule. Now, moving second property is that moving the vector from one organism to another should be relatively easy. Generating and purifying large amount of vector DNA should be straightforward. Mechanism to select host cell containing the vector. Ability to insert genes in the vector. Detect the presence of an inserted gene in the vector. So basically, we will learn in the next slide what are the different features of a cloning vector. So it must have an OD site. So OD site is the origin of replication. If it doesn't have this OD site, if it does not have this OD site, then this plasmid cannot replicate inside the bacterial cell. So I'm talking about plasmid vector now because it is most commonly used and it is easy to understand as well. So it must have an OD site. It must have multiple cloning sequences so that you can insert your gene of interest, right? Uh, in insert gene of interest inside this multiple cloning site, and it must have some selectable markers, right? So that it must have this ampicillin resistant gene, tetracycline resistant gene, so that you can screen for your gene of interest. So for example, if your gene of interest gets inserted in a site which is having a tetracycline resistant gene, then your tetracycline resistance property gets inactivated. Insertional inactivation is one of the concepts which you must have learnt in molecular biology. So then your cloned gene will be sensitive to tetracycline, right? Because it has lost that property. So these are the few. It must have OD site. It must have selectable markers. It must have a promoter sequence. It must have Terminator side, it must have selectable markers. These are the few things. So what are the key features? So, this figure is taken from one of the book, Molecular Biology book by P. Clark. So you can read what are the different features that must be present in cloning vector. It must have an antibiotic resistant gene. Okay, so it makes host bacteria resistant to the corresponding antibiotic, so that bacteria harboring the plasmid can be separated from bacteria without the plasmid. So when once you are growing the bacteria in an LB tube or LB media, then only those bacteria which are having that particular resistance will only grow. Another bacteria will not grow. The contamination is also removed if you are using this antibiotic in your media. Then there will be selectable markers. So any gene that makes a bacteria harboring the plasmid have an easily identified trait, which allows the bacteria that have the plasmid to be separated from those that do not have the plasmid. Then antibiotic resistance is also an example of selectable marker. Why you need promoter gene? So if you clone, if you once you have cloned your gene of interest inside the vector. Now, if you want to express that particular gene, you want to express the protein from that gene, then you will require the promoter, right? So that your promoter can bind to that your promoter region. For example, you have this T7 RNA polymerase, and you have the T7 promoter in your vector. So this T7 RNA polymerase can go and bind to your T7 promoter, and it can cause transcription of that gene so that your protein can be expressed. So origin of replication will be required so that this vector can replicate on its own. Okay, and multiple cloning sites so that you can insert your gene of interest, right? You can read all this. Then insert primer binding site should also be required if you want to. So you have this T7 promoter and a terminator. So accordingly, if you want to sequence that particular gene, then you can use T7 primer, forward primer, and T7 reverse primer to sequence that gene. Okay, then there should be terminator also, so that if you want to express the protein from the gene, then there must be a start site, there must be a stop site terminator. Okay, so all these things should be there. Now, what is the purpose of origin of replication in plasmid? So origin of replication, as I was mentioning, the plas so origin of replication purpose is to facilitate, provide antibiotic resistance. No, this is not the property. This is the function of antibiotic resistant genes or selectable markers. To express foreign genes, you need promoter. You need promoter to express foreign gene. To facilitate insertion of a foreign gene, you need multiple cloning site to allow plasmid to replicate independently. That is the function of OD. Okay, so option D is the correct answer. This is your selectable markers. Okay, so this question is clear. Now moving to the next question, the feature that is essential for selecting transformed host cell. Okay, so you want to select host cell. So basically what is done after the cloning is done, a transformation is done. That is what you do, you insert or you, once you have the cloned gene, so uh, this is your cloned gene of interest and this is your vector. Now you have to transfer it into a host cell, for example, into a bacteria. So this process is called transformation. Now, only those bacteria, for example, your this plasmid, this plasmid has ampicillin resistant gene. And now what will happen when you have done the transformation, you will plate it on an agar plate having this ampicillin antibiotic. Then only those bacteria which are having the ampicillin resistant gene, only those will grow, while others will not grow. So that is essential for what? Selection marker. For selecting transform host, you need what? You need selection markers. Answer C. Now there are these different bacterial cloning vector like PBR322, PUC19, PUC19. All these things were discussed in the lecture. So just quick revision. What are these vectors? So PBR322, where P indicates it is a plasmid and BR indicates the laboratory in which the vector was constructed. So it stands for Bolivar and Rodriguez, the two investigators who developed PBR 322, and 322 distinguishes this plasmid from others. So this is different. P is standing for your plasmid. BR stands for the laboratory which it was discovered and or designed or constructed, synthesized, not discovered. It was synthesized, and 322 stands for the strain number. So there are different types of plasmid: 322, 325, 327. Now this PBR 322, it has two different the antibiotic resistant gene. One is this ampicillin resistant gene, and another is tetracycline resistant gene. So and these this resistant gene also has several cloning sites. Multiple cloning sites are present in this resistant gene, so that you can screen whether your gene of interest has been cloned in this vector. So if it gets cloned in this particular sequence, then the bacteria will become sensitive to ampicillin. Similarly, if your insert is or gene of interest has been cloned in this BAMH1 site and SAL1 site, for example, then your bacteria will now become sensitive to tetracycline. So you can select for the bacteria which is having this your gene of interest. Okay. Um, so these things I think that we all have studied in class 12, that how we can screen. Okay, I'm not going into detail. It carries two set of antibiotic resistant genes, either ampicillin or tetracycline resistant, and can be used as a selectable marker for cells containing the plasmid. Now, PBR 322 has a reasonably high copy number. So all these are different features. It has two restriction anti-selectable markers, and it has a high copy number. All these things. Okay. Now we will go to alkaline lysis method. So how is plasmid? Now you want to purify the plasmid from your bacterial cells. So how is it done? So one of the method is by alkaline lysis method. Okay, so alkaline lysis method follows the principle of the configuration of a plasmid. Now the bacterial plasmid that is there, that it can have two different conformation. Not only two, it can have actually four different conformation. One is supercoiled, open circular also known as nicked circular. It can have circular, uh, closed circular conformation is also present, and there is a linear conformation which is also present. So basically the alkaline lysis method, what it follows is that it distinguishes the conformational differences between plasmid and bacterial DNA is used to separate two types of DNA. And most plasmid in the cell exists as a supercoiled molecule. So your genomic DNA. So when you are trying to isolate the plasmid, once you open the bacteria, the lyse the bacterial cell, your lysate will have both the bacterial genome and the plasmid also. Now you want to separate the plasmid. You do not want the bacterial genome. You only want the bacterial plasmid. So how can you purify the plasmid from the lysate having your genome? That is done on the basis of the conformation of the bacterial genome and the bacterial plasmid. The bacterial plasmid is supercoiled, while your bacterial genome is it is a large molecule compared to the plasmid, and uh, it does not exist as a supercoiled structure. Okay. You can have different conformation. It is relaxed covalently closed circular DNA. It can have a supercoiled DNA conformation, and it can be open circular DNA conformation. So I'll quickly explain what is the alkaline lysis method in this figure. I'll just create a slide. So basically in alkaline lysis method, you have three different solutions: solution 1, solution 2, and solution 3. Okay, the solution 1 is called the resuspension buffer. Okay, solution 1 is called the resuspension buffer. Solution 2 is called your lysis buffer, and solution 3 is called your neutralization buffer. Okay, neutralization buffer. So what the first step is, what you need to grow the bacteria. You need to grow the bacteria first of all. So you take a 10 ml culture, set up a 10 ml culture, you grow it for 16 hours. Once it has attained sufficient growth, then what you do, you pellet it down. You have the bacterial pellet. Once you have the bacterial pellet, you resuspend the bacterial pellet in your resuspension buffer. Bacterial pellet in the resuspension buffer. Now, what your resuspension buffer has, it has tris buffer, it has glucose, it has EDTA. Okay, tris, glucose, and EDTA, and also it has RNAs as well. So why are these components present? So what happens is when you lyse the cell, you will have your lysate will also contain DNases. Now the DNAs require magnesium, calcium, all these different divalent cations are required for the activity of the uh, different DNases. Now, if your DNase is present in your lysate, then what it can do? Your DNase will degrade your plasmid. So you do not want to degrade your plasmid. So you add the resuspension buffer. So the resuspension buffer contains EDTA. So EDTA will chelate out the divalent cations. Similarly, you have tris will provide the particular buffer, right? pH 7.4 or 7.5, and glucose will the osmolarity of the cells will be maintained. And what your RNAs will do, now you do not want RNA in your when you are isolating the plasmid, you only want the plasmid DNA. So that is why you add RNAs, so that the RNA will be digested. Next, you have the lysis buffer. So your lysis buffer, it contains zero point 0.2 normal NaOH and 1% SDS. Okay, so once you add the solution to the lysis buffer, what this will do, your SDS will digest the bacterial cell membrane, right? And NaOH, what it does, NaOH also helps in the lysis of bacterial cell membrane and the bacterial membrane proteins. The SDS will help in digesting your or solubilizing your bacterial membrane proteins. But another important function is performed by NaOH. What it does, NaOH will zero point 0.2 normal NaOH will separate the two strands of DNA. It will convert the double-stranded DNA into single-stranded DNA. It will denature the DNA. So once you add zero point 0.2 normal NaOH, it will denature both the chromosomal DNA and the plasmid DNA. Okay, so both your chromosomal DNA and your plasmid DNA will become denatured, will become denatured. Now, so at high pH, so at around pH 12.0, this will happen. Now, once you add neutralization buffer, your neutralization buffer contains potassium acetate and acetic acid. Acetic acid. So acetic acid and potassium acetate. Now once you add the neutralization buffer, it will lower the pH of the solution. Once the pH is lowered, what will happen? The DNA will try to renature. Now once the DNA tries to renature, both the chromosomal DNA will also try to renature, and your bacterial plasmid DNA, that will also try to renature. But the plasmid DNA will be able to renature because it is shorter in length. It has a small size, but your chromosomal DNA will fail to renature, and your it will form a precipitate. Your chromosomal DNA and all your proteins, bacterial proteins, they will form a precipitate, and you will see white colored precipitate like structure in your solution. Then after this step, what is done? It is centrifuged. It is centrifuged, and then you separate the the filtrate or the supernatant. You separate out the supernatant, and now after once you have got the supernatant, in the next step you do a phenol isoamyl alcohol chloroform treatment. Chloroform and isoamyl alcohol IMA. Isoamyl alcohol treatment. So once this treatment is done, then you will get you know this the protein will be all your digested protein will be separated in the organic phase, and your aqueous phase will have the DNA, will have your DNA. Now you get the DNA, you precipitate it with 100% ethanol, 100% 3-volume ethanol you can use, or you can use 70% isopropyl alcohol. So once you add this, your DNA will get precipitated, and you can separate out all these phenol, chloroform, and all the other reagents you have added previously. That then you can spin it down, and you can get the pure DNA. You wash it with 70% ethanol if you want, and then you can get your pure DNA. Okay, and then you can run it on an agarose gel and check. So you can read all this description. Okay, same thing has been written over here. You read this. So the basis is also, so that is what it is showing. This is again from the T.A. Brown book. So this is your linear DNA and your supercoiled plasmid. So once you see, you add the NaOH. So at high pH, your DNA is getting denatured. Okay, but this high pH, it will this supercoiled DNA will also get denatured, but it will not get denatured to the same extent because it is supercoiled. It will not get denatured to the same extent as your linear DNA. Now, once you lower the pH by adding glacial acetic acid and potassium acetate, which is your neutralization buffer, all your DNA, single-stranded DNA will get tangled and it will form a precipitate, while your this plasmid DNA will get denatured and it will remain in the supernatant. Now you do you spin your sample and you isolate the supernatant. Okay, and then you can do isoamyl alcohol, phenol, chloroform treatment, and followed by that, you can do this the aqueous phase. You can again treat it with 100% ethanol or 70% IPA and get your DNA. So here all the description is given. You can go through this slide. I'm not reading it out. Same thing is written. Now answer this question. The reagent used to denature plasmid DNA while keeping the chromosomal DNA denatured. What is this? Denatured plasmid DNA, it is your neutralization buffer. Buffer, isn't it? Option A. Renature the plasmid DNA because you are lowering the pH now. You are adding acetic acid and potassium acetate. Okay. Now coming to this bacteriophage lambda vectors. So this bacteriophage lambda vectors, what it has. So this is another type of vectors. So those were plasmid vectors is for the bacteria. These are bacterial vectors. Another you can use is bacteriophage lambda vectors. When is it used? If you want to clone an insert which is of higher length. So for if you're using plasmid vector, you cannot use inserts which are more than 8 kb you cannot use. Right. But if you want to clone genes which are having a fragment length of 25 kb, okay, then you can use bacteriophage lambda vectors. So this bacteriophage lambda vectors, so there's are so bacteriophage lambda is basically a bacterial virus that infects E. coli and it has been widely used as cloning vector. This virus, why is it used? It is easy to propagate and therefore has long been a model of organism. And in the lambda phage particle, the genome. So if you see the lambda phage DNA, it is a double-stranded DNA molecule, linear molecule. It is a linear molecule. The genome is linear DNA molecule and it has two cos sites. So this is very important. It has two cos sites. And why are these cos sites important? So after the phage injects its DNA into the bacterial host, the DNA circularizes. So if it does not have this cos sites, then the circularization will not take place. So for circularization after inserting into the bacterial cell, that is done by the cos site. So the two cohesive ends base pair and are ligated together by bacterial enzymes to form a circle. So after it enters the bacterial cell, so you see this figure circular after infection of bacterial cell, it has formed this. Uh, it has again circularized, but in the virus it is linear. It is linear. Okay, so the DNA molecules of between 37 and 52 kb can be stably packaged into the head of the lambda particle. So answer this question now. In the bacteriophage lambda, the cos sites are responsible for initiating transcription, no. Terminating transcription, no. Inducing lysis, no. Circularizing the genome. Okay, answer is option D. Transcription initiation will be done by promoter, termination transcription, the terminator, lysis induction, there will be separate gene that is for inducing lysogen. So circularizing the genome is the answer. Now what happens is that in this lambda phage vector, if you see the structure of the lambda phage DNA, so the two ends will have cross site, which is required for circularization that we just now understood. Other than that, it has a left end, a middle end, and a right end. So this middle portion of the DNA, that is non-essential region, and it is this region where we can clone our gene of interest. Okay, so in this middle region, our gene of interest is. So the left region has essential genes for structural proteins, and right hand region has genes for replication and lysis. But the middle region of the lambda genome is non-essential and may be replaced with approximately 23 kb of foreign DNA. Since the middle now, if you replace the middle region, so the middle region has one important function of integration and recombination. So once you inject this DNA into the host cell, for integrating into the host genome, this integrase and recombinase, these enzymes are required. But you have replaced it with your gene of interest, so this recombination can no longer take place, isn't it? So you will require helper phages. Okay, as the genes for such lambda replacement vectors cannot integrate into the host chromosome and form lysogens by themselves. To generate lysogens, it is necessary to use a helper phage to provide the integration and recombination function. Okay, so for this, you need to understand this concept. So this is. So what is done basically of packaging lambda replacement vector is you have a so the lambda cloning vector containing cloned DNA must be packaged in a phage head. So you have to package this your clone gene of interest inside the lambda phage head. Okay, so a culture. So what this is done is that a culture of E. coli is infected with a mutant lambda that lacks the gene for one of the head protein called E. Okay, one of the head protein. So for formation of the head protein, you require a protein E and you require a protein D. Now you have a mutant lambda phage vector which lacks the head protein E, and you have a mutant lambda plasmid which lacks the protein E, protein D. So you grow this E. coli culture separately. Okay, are grown with mutant lambdas, and the virus are induced to enter the lytic cycle. Now, although the E. coli are lysed by the phage, so the E. coli will be lysed by the phage. Now, in the lysate, you will have soluble mixture. So, you from the bacteria which is having this plasmid, which is having this lambda E plasmid, that will have all the different components of the phage particles, the tail, the hair, the tail and all the other different head regions will be present. But this E protein which is required for head packaging, that will be absent. And for here also the D protein will be absent. Now what you do is that the tails assembly protein, protein D is present, but it will no pre-heads due to lack of E. So the entire virus will not be formed because it lacks E. Similarly, here also the entire phage assembly cannot take place in the inside the bacteria because it lacks the protein D. Now you mix the two. You mix the two, and along with it you add the lambda DNA with insert. Now once you do this, you will have the lambda DNA with the insert, and now you also have the you have mixed this, so it will have both the protein D and protein E now, and now it can create this head, and the DNA can be inserted inside this head region of the lambda phage. You can go through this text, you will understand. These two lysates are mixed along with the lambda vector containing the cloned DNA. Although mixing is done in vitro, the components can self-assemble into a functional phage that can infect E. coli. Okay. So the type of vector that can insert larger fragments of DNA and includes cohesive site is what? So it is bacteriophage-based vectors. So bacteriophage-based vectors have cos sites, which is required for decircularization or required for circularization, not decircularization, circularization inside the bacterial cell. Okay. Now, what are shuttle vectors? Shuttle vectors are those that you can replicate, that can replicate in two different hosts. Why? How they can replicate in two different hosts? Because these shuttle vectors have two different origin of replication. Okay, it has origin of replication for bacteria, and it has yeast origin of replication, which is known as the ARS site, autonomously replicating site. Okay. So if you put this vector inside the yeast, then also it can clone, it can create copies of itself. If you put this vector in a bacteria, then also it can replicate on its own. And it also has antiviral, antibiotic resistant gene. It has multiple cloning site, and it will also have a centromere sequence. This is important. It will have a centromere sequence so that when this vector is inserted inside the yeast cell, then it can, when yeast division is taking place, then this centromere is required, right? Centromere is required, so it can be divided in the daughter cell. So one it will have two replication, origin of replication. It will have a yeast centromere sequence, and there will be selectable markers for both yeast and E. coli. Okay. Leucine biosynthesis gene for selection in yeast and antibiotic resistance gene. E. coli. Okay, now there is another concept about the yeast episomal vector. Yeast episomal vector is what? It is a type of shuttle vector only, because here you see it is having both origin of replication for bacteria and for yeast also. So the word episomal indicates that this yeast episomal plasmid vector can replicate as an independent plasmid, but also implies that integration into one of the yeast chromosome can occur. It can also be integrated into the yeast chromosome. So how does this integration occur? It is because the gene carried on the vector as a selectable marker is very similar to the mutant version of the gene present in the yeast chromosomal DNA. Okay, so you see this figure. So this yeast chromosomal DNA has the mutated leucine. So this I was explaining, right? Leucine biosynthesis gene for selection in yeast. Now this leucine biosynthesis gene is mutated in the yeast chromosome. Okay. And your yeast episomal vector will also have this leucine gene, but this leucine gene is normal. Okay. Now, because of this, what will happen? So because it is very similar. it is very similar. Homologous recombination can take place. So homologous recombination can occur between the plasmid leucine 2 gene and the yeast mutant leucine 2 gene, resulting in the insertion of the entire plasmid into one of the yeast chromosome. So you see here, because this plasmid, this is your yeast episomal plasmid vector, and it can this entire plasmid can be integrated into the yeast chromosome, which is having your mutated leucine gene. So the recombination between plasmid and chromosomal leucine 2 gene can integrate an yeast episomal plasmid 13. So after integration, what will happen? There are two copies of leucine 2 gene. Okay, you see here, once recombination has taken place, this gene has inserted into the plasmid gene, has got inserted between this mutated leucine 2. So there are two leucine 2 genes now. Okay, because this gene of interest by homologous recombination, it has inserted into your this plasmid vector. It has inserted between your leucine 2 gene by homologous recombination. So it has two leucine gene. Now one of this will be mutated and another will be functional. Other get is mutated. Okay, so that is what I was explaining. So you have the desired recombinant. So this is your desired recombinant, which has been inserted between your leucine gene. Okay, leucine gene, and this is your this is self ligation product that has formed, and this is also these are all self ligated product. This is also not having your desired recombinant. There has been some other part which has got, this is the self-ligated vector. And here this is some other non-desired gene of interest. It has got inserted into your yeast episomal vector. Now, if you transform, this is your sample, and you transform this into the E. coli, then there will be few E. coli which will you will get colonies which will be growing in ampicillin medium, and you will also get colonies which are having this which has this your correct desired recombinant gene. So you can isolate all these and you can grow them, and then you can sequence and see. So you purify the DNA from several clones, and then you identify the correct molecule. So the one which will have correct molecule, they will grow on both ampicillin medium and media which is not containing leucine. Okay, because this yeast has a gene which can biosynthesize leucine. So even if you do not provide leucine in the media, this yeast cell can grow. But these E. coli cells which are carrying the self-ligated vector or the undesired recombinant gene, then these E. coli cells will not grow. E. coli clones, bacteria cells, they will not grow, and only those which can grow in leucine-less medium, or medium which does not contain leucine, will be your correct thing. So this last question is for today is the type of yeast vector that is unstable and extra chromosomal is what? So it is answer is option B, yeast episomal vector. Okay, so these are unstable because they have this plasmid vector have leucine this has leucine. It can biosynthesize its own leucine, but your yeast vector or yeast chromosome, that that is mutated. That leucine is mutated. So that is why it will do homologous recombination and form this structure. So episomal yeast vectors are present as extra chromosomal DNA and are unstable. Okay. So this can overcome by integration of vector into the host chromosome. Your host chromosome, the yeast chromosome that you have, that lacks the leucine biosynthesis or mutated, the leucine biosynthesis gene is mutated in the host vector. So the in yeast integration occurs by homologous recombination. So in yeast integration, plasmid contains target sequence for integration into chromosomal DNA, selectable marker, and bacterial origin of replication. Okay, so you have the integration plasmid and you have a host chromosomal DNA. You have to understand this. Before vector delivery to the yeast, it is digested with unique restriction endonuclease to produce linear DNA to increase the transformation efficiency and integration. In most of the cases, integration is done in such a way that the yeast chromosomal DNA remained intact and integration may not affect yeast growth. So this is all that I explained you in the last two slides. Okay. So if you have understood, I hope that is all for today. So I will, if you have any more doubts, you can ask me, or else we can take up your questions in the next class as well. Okay, so I will end today's session now. Thank you for your kind attention. See you in the next class then.